Okay, earlier we had done some introductory material kind of talking about what anatomy students should learn and why we do comparative anatomy, and doing an overview of what the comparative anatomy course will consist of, which is not really any of that test material, right? So now we're going to get more into the actual study of comparative anatomy. We're going to begin with identifying what a chordate organism is and comparing it to the other taxa of animals. So what is a chordate in respect to the other animals? The first thing on here, chordate has 3 germ layers. That means it's not a sponge. Most other animals will have 3 germ layers: an ectoderm, a mesoderm, and an endoderm. Those 3 layers as you go from the outside to the inside. Ectoderm, mesoderm, endoderm. So sponges don't have that kind of organization, at least not well established. Next, chordates have bilateral symmetry. They have a left and right side. You can kind of divide down the middle and see that symmetry. So that's unlike cnidarians. Cnidarians are your jellyfishes, your sea anemones and hydras, those sorts of things that have a radial symmetry, so that you could kind of slice them in any direction around that center and you'd have a symmetrical kind of shape. So bilateral animals. Next, we have a tube gut. That's a gut that extends all the way through. It's not just a sac. In the cnidarians and in the flatworms, the gut, if it's present at all, is just a sac. The food comes in the mouth and then it's spit right back out. It doesn't form a complete tube through the body. Next, deuterostome. We'll talk more about this in a minute, but anyway, it has deutero means second, stome means mouth. So as the tube gut forms, if the second opening that develops in the formation of the tube, it starts at one end, goes through to the other end. If that second opening becomes the mouth, it's a deuterostome. If it's not a deuterostome, then the first opening becomes the mouth, and it's what we call a protostome. Examples of protostomes include arthropods, mollusks, and annelid worms. So, we've got 3 germ layers. We're bilateral. We have a tube gut. We're deuterostomes. And finally, we have a notochord. So that makes us not like the echinoderms. Echinoderm, that means spiny skin. So on the outside, they have more of a, it's not kind of like an exoskeleton, I guess, and that has a dermis that has these concretions in it that make them spiny skinned. But they do not have a notochord. So those echinoderms include things like starfish, sea urchin, sea cucumber. Those things are echinoderms. So these features, 3 germ layers, bilateral symmetry, tube gut, deuterostome, notochord, those are all features of chordates that help separate us from these other animal groups as we move through the chart here. Just to clarify something on this, the chart that I have listed here that separates the chordates from these other animal taxa is constructed in a specific way so that, first of all, sponges are considered the simplest form. They do not have any kind of body symmetry. They don't have their tissues organized into ectoderm, mesoderm, and endoderm like all of the other organisms. Then the next step up would be then to have 3 germ layers. So all of the cnidarians, the flatworms, the arthropods, mollusks, annelids, echinoderms, they all have that bilateral symmetry. And then the next level up from that would be to have the tube gut. So flatworms, cnidarians, and sponges do not have that tubular gut, but everything else down here does. The arthropods, mollusks, annelids, and echinoderms, as well as the chordates. And then beyond that, all deuterostomes have that second opening that develops in the formation of the tube gut to become the mouth. Everything that had a mouth above that in a tube gut was a protostome. So arthropods, mollusks, annelids, and even some of the other lesser phyla and the roundworms included in there, would have that protostome condition. Echinoderms and chordates both are deuterostomes, but echinoderms do not have the notochord. So we could construct kind of a phylogenetic graph from this information if we wanted to. Next, we need to go into looking at some detail on how the deuterostome condition develops, and we want to talk about another factor we call the body cavity state. Some animals are considered acoelomate, meaning they don't have a body cavity. Some are considered pseudocoelomate, and we'll talk about why that is different from the more common condition of the higher taxa, which have what we call the eucoelomate condition. That means true body cavity. So acoelomate, no body cavity; pseudocoelomate, false body cavity; and then eucoelomate, true body cavity. So that's what we'll look at next. Okay, a little bit about this protostome, deuterostome concept. We need to look back at how the tube gut forms to understand this. So I've gone even further back here. This here is your one cell stage up here, a single cell. Then it divides into the 2-cell stage, and then there's a 4-cell stage, and so on. And these cells keep dividing to form the early embryo. Eventually, this will form a solid little clump of more cells, and within that clump, a space will start to develop. A cavity will form on the inside. So here's our cavity right here. Now, eventually, we're going to know that as a blastocoel cavity, but for now, just know there's a cavity in there. And these cells keep dividing. So you've got a hollow ball of cells, and in there you have that cavity. As they keep dividing, eventually, in some forms of development anyway, this is kind of how it happens. This is like the amphioxus, develops this way. On one side, there's a caving in of cells. The cells start to invaginate on one side. So we lose that nice spherical shape. It starts to cave in on one side. And these cells here, around where it starts to cave in, are going to form the inside layer of the tube gut, and those around the outside will form the ectoderm layer. So we're going to get endoderm and ectoderm formed by this process. So that's the initial formation of what we call a gastrula. Gastrula, gastric refers to stomach, so here's the beginning of the gut formation. So here's that opening later on as the animal continues to develop. That folding in continues to move through the organism from one end toward the other. And this opening right here is referred to as a blastopore. Blastopore opening at that end. And then as it continues to develop, and this moves all the way through to form the tube gut, we'll end up with that solid endoderm lining along here, around the tube, and then around the outside of the animal we'll have the ectodermal layer. So we have endoderm and ectoderm. We don't have any mesoderm drawn into this picture. It's not that important to us at this point. But anyway, like we said, if this first opening that formed right here, that blastopore opening, if that's the mouth, then it's a protostome. If the other end over here, this opening over here, becomes the mouth, it's a deuterostome. Okay, so deuterostome. The word means second mouth. It refers to the second opening becoming the mouth. If on the other end, this end had become the mouth, and food was to move this way through the animal instead of that way, this would be a protostome animal. Okay? Proto, first, stome, mouth. So it's not a protostome, it's a deuterostome if it's a chordate animal. Okay, next a word about coelom states in animals. Coelom, C-O-E-L-O-M, coelom, refers to any kind of cavity, so has a more generic meaning, but here specifically we're talking about coelomic states. We'll be looking at these examples. The first example is from a flatworm. I didn't draw it flattened, but you could imagine this if this were squished down. We have the 3 germ layers. We have ectoderm, I'll just abbreviate that, ectoderm. On the inside we have the endoderm. So those are the outside and inside that we'd already talked about. And then between the 2 layers we have some tissues here we call mesoderm. Mesoderm. So if the entire space between the endoderm and the ectoderm is filled with mesoderm in there, there's no body cavity, there's no space inside the body other than the gut tube in there. And that's not what we're talking about when we talk about a coelom. We're not talking about the gut tube. We're talking about between the gut and the outer body wall, that there's a cavity in there that holds organs and so forth. So anyway, in this animal, the organs are in there, but they're just stuffed in to all that mesoderm and are part of that region here. So that's, like I say, in a flatworm type animal. Later, when you get into the roundworms, you get this pseudocoelom condition. So we have the ectoderm, we have the endoderm, we have the mesoderm, but the mesoderm is only found out here adjacent to that outer ectodermal layer. And since there's not another layer of mesoderm around the gut here, that's why we call it a pseudocoelom. So your pseudocoelomates do not have a layer of mesoderm adjacent to the gut. They've got this cavity in here between the gut endoderm and the mesoderm out against the body wall. In a typical roundworm, that mesoderm would be mostly muscle, so it allows the movement of a roundworm. So this was the acoelomate up here, like a platyhelminth flatworm. Next we have the pseudocoelom, as we see in the roundworms. And then finally the eucoelomate condition. A eucoelomate, meaning true cavity, has the mesoderm both out here, so we have an outer layer of mesoderm to the outside. We have the true coelom there. Then we have another layer of mesoderm. So we have mesoderm, cavity, and then more mesoderm before we get to the gut. So this space between the mesoderm next to the gut and the mesoderm next to the body wall is the true coelom, the body cavity, the coelom of the animal. And you recall from our chart, once we get beyond the flatworms, most everything was a coelomate. There are a few phyla, lesser phyla, and the roundworms that have this pseudocoelomate condition, but most of our higher phyla are of a eucoelomate condition. That would include the mollusks, annelids, arthropods, echinoderms, and chordates. All have this layer of mesoderm not only against the outer body wall, but also next to the gut. And typically there's some kind of a membrane or connection between the outer body mesoderm and the inner mesoderm in here. Some kind of a mesentery, a connecting sheet. And we'll talk more about this when we talk about embryology later on in the class. But this helps to kind of explain what's going on with these different terms regarding the body cavity or coelom. Acoelomate, pseudocoelomate, and eucoelomate organisms. This is considered more primitive, this is kind of intermediate, and that's more advanced, supposedly. Okay? In our last talk, we had identified some of the features that separate the chordate groups from other animal phyla that we know of, and we had a list there. Now we're going to talk a little bit more about what makes a chordate different from the other phyla. We identified that the notochord was the primary feature there, but there are a couple of others. Some people say 3 different characters, some say 4. We've already mentioned notochord. There's also a neural tube, the extension of the, well, in humans it's the brain's spinal cord, extends down from the brain, the nerve tube. We just call it a dorsal hollow nerve cord. So that's the second. The third is pharyngeal gill slits. Although that might be better described as an arrangement of pharyngeal arches, was what I would prefer instead of gill slits, pharyngeal arches. So those are the three that I will be requiring that you be familiar with as those characters that set the chordates apart from other animal taxa. Now there's a fourth one that some people include, it's the post-anal tail. So there's an extension of the body beyond the end of the gut. So the gut goes so far and then the post-anal tail beyond that. But we want you to know these three for your comparative anatomy study. Notochord, dorsal hollow nerve tube or cord, and pharyngeal arches. Now let's take a look at what the basic body plan for all chordates would be based on that arrangement. Let's say we have a body, this is kind of just a hypothetical kind of body structure for a chordate body plan. Here's the gut tube that we talked about earlier when we talked about the deuterostome condition. Remember that the gut begins here, and as it goes through the embryo, it comes out to the other side where the mouth will be in a deuterostome. So there's the overall body. On the outside we have ectoderm, in the gut tube we have endoderm, and between the two we have some mesoderm. Coming from the ectoderm right along the middle of the dorsal part of the body right here is the beginnings of the nerve cord here. So in blue we have the nerve cord going through here. What we think in humans as the spinal cord there, and brain would be the front end. And then directly underneath of that, a special feature. That's the notochord we talked about. So the nerve cord is actually derived from the surface ectoderm. The notochord is induced to form just beneath that nerve cord in the mesoderm of the body, right along the middle there. So there's our first two, our notochord and our dorsal hollow nerve cord above the notochord, and then pharyngeal gill slits. So here's our gut tube, but the very front part of this gut is going to be the pharynx. And around that pharynx, we're going to have structures called pharyngeal arches. Now in fish, they look something like this, but in the amphioxus and the tunicate that we look at, we're going to see something a little different. But anyway, a structural arrangement around the outside of the pharynx that in the early forms allows water to move from the pharynx to a space outside of the pharynx or to the outside of the body in fishes. So this is the basic plan for all chordate organisms. You have the ectoderm, endoderm, and mesoderm, and then from the ectoderm you get the dorsal nerve tube to form, and then beneath that the notochord develops, and then around the pharynx in the front you get some kind of a structure we can refer to as pharyngeal arches, sometimes called a pharyngeal basket. Anyhow, That's the very beginnings of a core date.